Okay, I'm gonna go ahead and get started. Well, if you're new to the ESTIV and ASCCT webinar series, my name is Christy Sullivan. I'm the secretary of ASCCT, and welcome to the first webinar of 2023 in this series, which is usually a monthly webinar. Happy New Year, everyone. I encourage you to visit our websites to find out more about our organizations and the activities we undertake, which include scientific meetings and webinar series like this one. And in particular, one of the initiatives your membership supports is to provide support for students and early career scientists during both organizations' annual meetings. And so we provide awards for presentations for students and early career scientists, and then we give them the opportunity to present their work to us in our webinar program. So we have very exciting work to see today from two of our award winners. First of all, I just want to give you a couple of housekeeping notes. The webinar is recorded, and the recording will be posted on the ASCCT website a few days from now. It usually takes us a bit of time to get it up there. In the webinar platform, you can ask questions of the presenters using the Q&A feature at the toolbar at the bottom of your screen. Today we'll have separate Q&As, so each person will give a presentation, and then we'll have a Q&A, and then we'll have the second presentation and Q&A. So get your questions in for the speakers while they're talking. So again, please put your questions in the Q&A. If you have any technical issues or comments, you can put those in the chat. We've also enabled closed captioning subtitles. If you want to view or hide them, you can do that at the bottom of your screen where you see a live transcript or in your toolbar. And finally, I hope you will say hello in the chat and let us know where you're joining from. I just wanted to give you some information about our next webinar. It is on Wednesday, February 22nd. And it's going to feature 3 speakers, again, from our annual meeting, giving more details and information, going into more in depth about their work and presentations that won awards at the meeting. So you can see there's a nice breadth of presentations that will be coming in February's webinar. And so I hope you'll join us. And you can register, at the website address at the bottom there. And so let me then go ahead and introduce our 2 speakers for today. So first of all, we have Amber Daniel from Inotiv. She's a toxicologist in the Predictive Toxicology and Information Sciences group. She holds a Bachelor of Science in Animal Science and a Master of Toxicology degree from North Carolina State University. And as a contractor supporting NICEDM, she's worked on a variety of projects to promote the development, use, and regulatory acceptance of alternatives to animal use for chemical safety testing, including validation studies of in vitro methods and defined approaches to detect potential chemical safety hazards. And additionally, which is what she's going to be talking about today, she provides support for Microphysiological Systems for COVID Research working group. And her work at the annual meeting won a Best Flash Poster Award. I'll introduce our second speaker, and then I'll turn the floor over to Amber. Our second speaker is Sunggun Lee, a student at Duke University. He is a junior studying biomedical engineering and electrical and computer engineering. He's interested in projects related to health equity and international development through designing tools that are low cost and accessible. Sunggun is involved in organizations and clubs such as Duke Engineers for International Development and Engineering World Health. He's also an editor for the school's official student-led engineering publication, the Duke Engineer magazine. He's worked on several projects in machine learning, including in the Gong Lab at Duke University for the segmentation of neurons, and for GAMI, a nonprofit organization to develop a mobile application for accessible anemia diagnosis. Very excited to hear about Sunggun's work on deep learning for toxicity models. And with that, I will stop sharing my screen and turn the floor over to Amber.
Hi, everyone. Thank you, Christy, for the introduction and for the invitation to speak today. I'm really happy to be sharing some information about MPSCoRe with you all. My role with this group is acting as a liaison with leadership for organization and coordination of efforts of the working group and dissemination of relevant information to the members, and really just acting as a central point of communications. I've had a lot of fun working with this group. Not being an expert on microphysiological systems myself, it's been a really great learning opportunity for me, and it's become one of my favorite projects, and I'm really excited to be able to share some of that with you all. All right, so today I'm just going to give some background information about the working group and its objectives and highlight a few of the efforts that MPSCoRe has supported or is currently supporting as examples of how they've addressed those objectives so far. And I'll also touch on some of the activities that we're planning for the year ahead. My goal for today is that you'll walk away from this webinar with a good understanding of what MPSCoRe is and how and why it was established. And I especially hope that you'll all be able to appreciate the potential impact that this working group has in accelerating the modernization of infectious disease research, and how in turn that can help to not only improve human health, but also reduce the use of animal models in this space. So the emergence and spread of COVID-19 really put a spotlight on the need for more effective approaches to prevent, treat, and control the spread of infectious diseases. Animal studies have historically been used for this type of research, but it's pretty well recognized that animal models have some limitations, including their biological relevance to humans. Susceptibility to disease and manifestation of symptoms in animals can, of course, be quite different from what's typically seen in humans. So the pandemic and the public health concerns surrounding it really provided researchers with a unique opportunity to work on the development and assessment of in vitro platforms that are human cell-based and that may be more appropriate compared to those animal models for mimicking the susceptibility and effects of COVID in human lungs or other human organs. And these in vitro platforms are called microphysiological systems. I'll be referring to them as MPS today. Some of you may know them as organ on a chip. In having the global scientific community working rapidly and concurrently to develop these MPS, some challenges became apparent. So for example, end users need to understand the utility and the context of use of the different models that are available so that they can choose the model that best suits their specific needs. But with that number of available models increasing so quickly, it becomes increasingly difficult to maintain that high level of awareness of what's available. Also, if there's no central body coordinating the widespread development of MPS for COVID, then there's a high risk of duplicating research efforts and not getting the best use of the available resources. And so there was this need for a neutral forum where we could bring together key stakeholders from across the world. And when I say stakeholders, I'm referring largely to researchers, regulators, model developers, and pharmaceutical manufacturers, to facilitate those connections and communication between them and to support them in overcoming these challenges. And so that's what MPSCoRe does. Really simply put, it's a network that connects individuals from these four sectors and opens pipelines of communications concerning the use of MPS for COVID-19 research. MPSCoRe was organized in early 2021 through a partnership of the National Center for the Replacement, Refinement, and Reduction of Animals in Research, which is based in the UK. My group, the NICEDM group within NIEHS, NCATS, the NIAID Division of Microbiology and Infectious Diseases, and the US Army Development Command Chemical Biological Center. And we currently have individuals from each of these providing leadership as board members, including our co-chairs Anthony Holmes, who is the Director of Science and Technology at NC3Rs, and Nicole Kleinsteuer, who is the Director of NICEDM. So these next few slides are really just kind of to illustrate the diversity of the different affiliations that are represented in the MPSCoRe membership. So this is the government and regulatory sector, MPS developers, drug and vaccine manufacturers, and then academia and MPS networks make up the largest portion of MPSCoRe. This actually takes up 2 slides. So in total, we have about 130 individuals on our roster from about 70 different affiliations. The overarching goal of MPSCoRe is to maximize the scientific, human health, and animal welfare impacts that MPS can potentially offer. And we have 6 objectives that define that goal. And for the rest of this presentation, I'd like to highlight some of the activities that MPSCoRe is supporting or leading to just kind of show how they've been addressing these objectives so far. One of the earliest efforts was identifying the members' biggest challenges. So what you see here, this is a figure from a publication that was authored by the MPSCoRe co-chairs that describes basically the establishment of MPSCoRe. So we surveyed the membership and asked them to identify their top 3 scientific and practical challenges that were affecting the use of MPS for COVID research. And some of the more common issues are illustrated here. The biggest scientific challenges were incorporating innate and adaptive immune responses in the models, accessing normal and diseased human tissue, staying aware of what MPS models are available, as I mentioned earlier, et cetera. And then the biggest practical challenge were things like securing funding, access to higher biosafety level facilities, labor and cost concerns, and so on. And so this kind of shows that these challenges exist, that there are a lot of them. And also this information has been really useful for leadership in that it's helped to kind of spark ideas and to guide our activities to the areas where the stakeholders need the most support. We encourage members to share data from their MPS studies whenever possible. Sharing data supports 2 objectives, which are connecting the technology developers and end users and characterizing criteria for model performance and readiness. So this graphic shows the content of the Biostatistics Analytics platform, formerly known as the Microphysiology Systems Database, that was developed by the University of Pittsburgh Drug Discovery Institute. It's a web-based resource that can be used to access, share, and model high complexity data sets to predict biological outcomes. And MPSCoRe actually funded the expansion of the Biostatistics platform to include a portal that's devoted to COVID-19. This portal was designed by Mark Sherdak and Bert Goff from UPitt. It was designed to be a central repository where MPSCoRe members can upload and share their data and where they can easily access other COVID relevant information and resources. So I've included some screenshots just to kind of give a general idea of how the COVID-19 portal is organized. So this is the landing page of the platform. You should hopefully see the URL down on the bottom left of the screen. And then the COVID-19 portal can be accessed from the diseases tab at the very top. Within the portal, information is organized into 6 main categories. And if you look below the light blue box with the page description, you'll see there's a second level of organization, and you can just toggle between those subtabs to see the resources that are available under each. So I'll just quickly scan through just so you can get kind of a feel for it. Notice that the entries are all hyperlinked, so you have really quick and easy access. The components tab is really useful. It can help with finding reagents, viruses, proteins, antibodies, and other materials. I just wanted to highlight the induced pluripotent stem cells portion, which gives users detailed information so they can quickly identify what is a good iPSC sample or iPSC derived cell for my model, where do I get them, and how do I use them. And there's a green bottom at the bottom of each page where you can suggest additional entries if maybe you're aware of something, another resource that exists, but you didn't see it included. And then one last feature of the portal that I wanted to mention is that MPSCoRe members with a registered account can privately share their own information and also access the profiles of other MPSCoRe members. So it's a really great venue for stakeholders to make connections. Another key objective of MPSCoRe is the assessment of MPS models against concurrently generated preclinical and clinical data. And MPSCoRe has supported this objective by partially funding the establishment of MPS technology for testing COVID therapeutics and vaccines at the NIAID Integrated Research Facility. So they're running a proof of concept study that uses MPS models to test the safety and efficacy of novel COVID vaccines and therapeutics, which then enables kind of a parallel side-by-side evaluation to compare data that are generated from models of these various complexities and assess the added value of the tissue chip. So we've heard recently they're in the process of completing comparisons of the different COVID variants in a lower airway MPS model, and they have just a couple of outstanding experiments left to conduct before they can compile the results. As a positive control to the COVID experiments, they've also been using MPS to evaluate a couple of small molecule antiviral treatments for a different virus. It's called the Nipah virus, which also affects the respiratory tract. And we've heard that so far those data look promising. They're also optimizing a flow cytometry panel for staining neutrophils run through infected chips to achieve an immune response. And they're working on validating procedures of inactivation of chips out of biocontainment and how best to test complete inactivation of the virus. So they have a lot of exciting work going on, and we're really looking forward to hearing an update on this from them within the next few months. Something else that we do is we hold regular virtual events to facilitate stakeholder engagement. We've had a couple of workshops that were focused on discussing different MPS models for testing therapeutics and understanding disease mechanisms. We've held several webinars where MPSCoRe members volunteered to present their research, and that led to some, really robust discussions. We maintain an email contact list so we can broadly distribute relevant information, such as newly published literature, funding opportunities, and other important announcements for different meetings and things like that. Additionally, at the emergence of the Omicron variant, we held an urgent meeting of MPSCoRe membership with the World Health Organization, and we facilitated a quick response method for members to communicate their individual research capabilities and things that they need to conduct studies to gain a better understanding of the biology and the potential impact of the variant. And so it was really kind of neat to see this network of different groups around the world quickly respond and match the urgency of that situation. So currently, MPSCoRe leadership is in discussion of next steps for 2023 and beyond. That includes the possibility of expanding the working group scope to include other emerging infectious diseases, and also brainstorming ways to help advance regulatory acceptance of MPS approaches. We held a webinar this past summer where a colleague gave a talk about the European Union perspective on developing performance standards for MPS. And during her presentation, she reviewed the European Medicines Agency, or EMA's, process for submitting and evaluating a non-animal testing approach for regulatory acceptance. And she mentioned that EMA has a scientific, regulatory, and legal multidisciplinary group that's called the Innovation Task Force, which offers an informal discussion platform for exchanging information and for provision of guidance to applicants that are early in their development process. And one focus of the Innovation Task Force is regulatory acceptance of new approach methodologies to replace the use of animals in testing of medicines. And the objective is to encourage development of these methodologies and accelerate their integration in the regulatory framework. But the Innovation Task Force had seen only a few such methods submitted, and as of that time, none had been successfully integrated into their regulatory framework. So MPSCoRe leadership discussed this, and in keeping with the working group's objective to work with regulators to better understand regulatory needs and decision contexts, it was proposed that we aim to hold a symposium workshop event in 2023. And we're still in the preliminary stages of planning that, but it seems likely that it will be a 2 half-day virtual event in the late spring. We're hoping we can feature presentations on MPS models for infectious disease research and where they are in terms of readiness. And we would also like to feature presentations from international regulators to kind of spark discussion and collaboration about their current regulatory approaches, and really just to raise awareness of the opportunities like those offered through EMA. And then the outcomes of this symposium and research would hopefully help establish a more concrete path to move MPS models from research to readiness. And then lastly, I just wanted to highlight just a couple of specific examples of the impact of MPS relevant work. So this is from an article that was co-authored by a few members of MPSCoRe that presents a literature survey of primary research that used MPS to study COVID-19 brain cell infection and effects of COVID on the human brain. And in summarizing the main findings, first, there were some groundbreaking findings, and several different groups were actually able to reproduce results of that were in different types of human brain organoids, and that mirrored some of the neurological symptoms that were reported in clinical studies. And so this article suggests that MPS models may be instrumental in confirming some of the hypothesized key mechanisms of neuro-COVID. Additionally, a different group at the Harvard Vice Institute used this human airway chip model to look at FDA-approved drugs, and contrary to findings of more standard in vitro animal cell-based systems, their results demonstrated a lack of efficacy of chloroquine, and that was consistent with the results of human clinical data. So work in this human MPS system has been used to support efforts to repurpose the anti-malarial drug amodiaquine for COVID treatment. And so I just want to reiterate that MPS are human relevant in vitro platforms that can be used to study infectious diseases and to develop effective vaccines and therapeutics. The MPSCoRe working group was established as a neutral forum to connect key stakeholders and coordinate global MPS efforts to study COVID-19 through activities that are guided by their 6 objectives. These efforts will accelerate the development and adoption of MPS in infectious disease research, resulting in the improvement of human health and a reduction in our reliance on animal models. And so I'll just end by acknowledging again the MPSCoRe board members and support staff and others that are leading the activities that I discussed today, not only for their efforts in this working group, but also for their very valuable feedback on the abstract and poster. And we would also really appreciate all of the MPSCoRe members for their participation in the working group. If you are interested in learning more about MPSCoRe or becoming a member, please feel free to reach out to me via email. Please also, if you know someone else who may be interested, feel free to nominate them and share my contact information. And I thank you all very much for your attention today, and I'm happy to take any questions.
Thanks, Amber. That was a great presentation. I have to say, I came up with a question and then you answered it. And then I came up with another question and you answered it. So it was a very clear, comprehensive presentation. While we're waiting to see if anyone wants to add a question to the Q&A, let me ask you another one. So I noticed that you mentioned some interest in looking at other risks or other infectious diseases. Mm-hmm. That kind of, I was thinking, you know, there's a lot of reporting around the interaction between COVID-19 infection and RSV infection, and I'm wondering if you've had any discussions about that particular, interaction and considering some studies with the MPS systems.
Yeah, no, so right now we're still very much focused on COVID-19. We have actually not had a discussion on that specific interaction so far. We're kind of focused on, the regulatory aspect a little bit first, I think, and then maybe later in the year might be moving into those, more incorporating different emerging infectious diseases. But I can certainly pass that on and, to the board and the MPSCoRe members and see if there's any interest in that.
Great. And maybe one other suggestion, and maybe you're already aware of this, is I know that some folks at NICEDM are involved in the COVID AOPs project. And I think this would be a fantastic way for both projects to validate each other in a way, having MPS systems generate some data to help improve our knowledge of these COVID pathways and how they result in disease, and also having the AOPs that are being developed come back, to kind of support method development or endpoint detection or endpoint implementation, or kind of the improving the MPS systems.
Right. Yeah. No, that sounds like a great idea too. I'm sure that's been discussed between the two groups. Yeah.
Great. So I don't see any questions. If you do have questions that you think of in the next couple of minutes, go ahead and pop them into the Q&A, and Amber can answer them in writing. But I think we'll go ahead and move on to Sunggun Lee, our next presenter.
Hello, my name
Good.
Hello, my name is Sunggun Lee, and my work is titled Applying Deep Learning Toxicity Models Across the Chemical Universe. The views or opinions expressed in this presentation do not necessarily represent the views or policies of the NIHS or the DNTP. So, chemicals are found everywhere in the environment, medicines, consumer products, and many other exposure routes in day-to-day life. So it's important that toxicity testing guides maximally informed decisions on chemical use and regulation to best protect human health. Traditional carcinogenicity testing generally relies on in vivo or in vitro experiments with a heavy emphasis on animal testing. And carcinogenicity assessments are usually directed by established guidelines, one of which is shown here, which is the ICH guideline on the need for carcinogenicity studies for pharmaceuticals, which formulates an evaluation process for human carcinogenic risk of chemicals. The ICH guideline was established with the intention to support an approach that reduces the use of animals in accordance with the 3Rs principles, which is an improvement from the previous S1B carcinogenicity assessment guideline, which was established by the FDA. However, the general experimental approach still remains very demanding, requiring a long-term carcinogenicity study in the rodent, which is about 104 weeks, along with one other study that supplements the main study that can either be a second long-term study or a shorter study of 29 weeks. And these general studies, whichever pathway you take, on average requires around 500 rodents and costs over $1 million. And so these studies are usually only conducted when human exposure warrants the need for them. And so essentially, carcinogenicity studies operate in a reactive way rather than a preventative way, and are time-consuming, resource-intensive, and frequently results in flawed generalizations to humans. Therefore, there's a need for alternative approaches of carcinogenicity assessment. In an increasingly data-driven world, new approaches that explore in silico methodologies such as QSAR models and adverse outcome pathways are emerging as attractive alternatives to traditional toxicity testing, where sufficient data is available. And programs such as Horizon 2020, Tox21, and other partnerships have supported this by providing large, well-informed data sets to encourage the development of these alternative approaches that are less reliant on animal testing. These in silico approaches have been successful, such as QSAR models that provide an efficient evaluation of the toxicity of a molecule using only its chemical structure. And so I put a paper here, this is just one example, like groups such as Zhang and others, they built a QSAR model that covered a wide range of chemical classes based on existing data from rat carcinogenicity with a great overall accuracy. It was like around 0.9 or so, which is very good. In particular, among the in silico approaches, the deep learning approach is a relatively newer and superior approach for carcinogenicity assessment. The application of conventional QSAR models are generally limited to more specific domains and are difficult to generate from random and diverse databases, which is usually the case that we're working with. And so deep learning models are a beneficial alternative because of its ability to parse large, intricate data sets and predict complex endpoints. And so this project involved the use of two deep learning models, DeepCarc and DeepDILI, developed by collaborators at the FDA to predict chemical endpoints for carcinogenicity and drug-induced liver injury, respectively. These models have been shown to outperform conventional QSAR models and state-of-the-art ensemble methods, and they're open source, and the GitHub repos are linked below on the right bottom. And in this study, the DeepCarc and DeepDILI were applied to predict toxicity for over 7,000 compounds in the Tox21 dataset, which is composed of food additives, consumer products, and more. So the general workflow of the DeepCarc and DeepDILI models is diagrammed on the left, with both incorporating an ensemble approach of 5 conventional machine learning algorithms integrated into a neural network to generate probabilistic predictions for carcinogenicity and liver injury. What this means is instead of using one machine learning model to make predictions, we're using 5 different models from which we choose the most optimized models. And using more models gives us the opportunity to obtain a better predictive performance compared to using one model alone. The selection strategy of the base classifiers were different for each deep learning model, with the DeepDILI model using an original selection strategy that selects optimized base classifiers from a rank ordering system, while the DeepCarc model uses a supervised selection strategy, which is based on more of a threshold system. Additionally, in order for the models to learn and obtain information from the compounds have to be represented quantitatively to the models. So there are chemical descriptors such as MOL2 and MOL2VEC, which can be thought of as a numerical label that contains the chemical characteristics and information of the compound. And from this, the model can learn and generate patterns. So as you can see in the diagram, in the first step, from the training set of the compounds, they're converted into a quantitative representation in which the models can learn from. Both the DeepCarc and DeepDILI model outputs probabilistic predictions of the carcinogenicity or liver injury potential of a compound, respectively. This means a probability output closer to 0 is considered low risk, while a probability output closer to 1 is a high risk compound. So in figure 3a, we observe the probability distributions for carcinogenicity of the Tox21 dataset. We can observe that there's generally a low carcinogenicity risk of the compounds, with around 53.2% of the compounds with low risk having a probability output of less than 0.1. But on the other hand, the probability distribution for drug-induced liver injury of the Tox21 dataset in figure 3b suggests that there's generally a higher risk, with 58.8% of the compounds predicted to cause liver injury, and even 24.7% of the compounds with a high risk, that is a probability output greater than 0.9. Figure 4 examines the high-risk compounds with greater than 0.9 probability for carcinogenicity and liver injury using the Integrated Chemical Environment Tool, ICE, developed by the National Toxicology Program. In particular, the molecular weight and the octanol to water partition coefficient, which is basically an indication of how hydrophobic the molecule is, were examined. And these are aspects of Lipinski's rule of 5 describing characteristics of an orally active molecule. And it's important to note that there are other properties, and the rule of 5 itself does not capture the complete story of the activity of a molecule, but it still provides a perspective from which we can examine the compounds. And from the figure, we notice that the molecular weight of high carcinogenicity risk compounds are generally significantly lower than the entire TOX21 compound molecular weight. And the log of the partition coefficient is in a tighter range, suggesting that the compounds are neither too hydrophobic or hydrophilic. And these characteristics indicate that the compounds that are high risk for carcinogenicity generally have good permeability and uptake in the human body. In figures 4C and 4D, we see that the molecular weight of the high liver injury risk chemicals tend to be greater, and the log of the partition coefficient tends to be larger, suggesting that these compounds are generally more hydrophobic. And to further examine these high-risk compounds, and especially how they exist in the consumer use, we used data from the US EPA's Chemical and Products database. It was fascinating to see that many of these high-risk compounds were actually not found in the Chemical and Products database, with 79.5% of the high-risk compounds in deep carc absent and 91.5% of high-risk compounds in deep DILI absent from this database. And for the compounds that were included in the database, in figure 5, we can observe that most of these high-risk compounds were found in consumer goods, such as personal care, appliances, and the construction and maintenance categories. And some of these high-risk compounds were prevalent in a variety of consumer use, such as 2-phenoxyethanol in 90 different uses as an antiseptic or preservative, and methylparaben in 87 different consumer uses as a preservative. And both of these lack conclusive evidence on human toxicity and yet are continued to be used. Additionally, visualization of the high-risk chemicals against approved drugs in the drug bank map was done using ChemMaps. ChemMaps was developed by Dr. Burrell, and it uses a complex assortment of 1D, 2D, and 3D pre-computed molecular descriptors to represent a compound in this 3D space. Compounds that have similar chemical characteristics are represented locationally closer to one another. The green chemicals are drugs that are approved, while the high-risk compounds are displayed as gray rockets. For the visualization of the high-risk carcinogenicity compounds on the left, we can observe a clear distinction between the spaces for the approved drugs and the high-risk chemicals. But for the high-risk liver injury compounds, there's more of an overlap with the approved drugs. And this suggests that a certain level of oral consumption of even approved drugs may cause liver injury, which is known. The main takeaways from this study is that with the availability of large data sets, such as the TOX21 dataset, in silico methodologies, especially deep learning models for toxicity predictions, can be used as a supplement or a guiding hand for targeted toxicity testing. Results of applying the deep carc and deep DILI models on TOX21 show that there's great power in the ability to assign numerical probabilistic toxicity outputs to the compounds. And from our analysis of the high-risk compounds, we observed that there's further research that may need to be done and conducted on some chemicals that are still widely used by consumers. And this study will be continued by applying the models to other data sets, such as the EPA's DSS Tox, which contains around 1 million compounds. And we'll conduct analysis of these results as well. And moving forward, we also want to improve upon the deep carc and deep DILI models by further refining them by implementing different chemical descriptors and beyond. Thank you for listening to my presentation, and I'd be happy to take any questions you have.
Thank you, Sunggun. That was a really nice presentation. I learned a lot about your model. We have a couple of questions. First of all, yes, to Andrea in the chat. The slides will be put up on the website as well, I believe. But we also have a question from Ted Simon, and he's wondering, can deep carc predict points of departure as well as hazard? And if so, could one do IVIVE and compare the deep carc points of departure to traditional cancer points of departure from animal testing?
Um,
You see
I
that
did
in the
not,
chat? Because it's kind of a two-part question. So
yes. I do not know what a point of departure is or an IVE. So if you could explain that to me, I think that would help me answer the question.
yeah, definitely. So so a hazard endpoint is basically a yes or no, right? There's a concern or not a concern. Whereas a point of departure would be, is there you know, a dose level or a level of exposure of the chemical that is of concern, or is sort of the level of exposure that you would have a concern for toxicity?
That's a good question. So the DeepCarc and Deep DILI models were developed on the NCTR liver cancer database and the DILIS database, respectively. And so whatever levels of toxicity that are found from the models depends on what were used in those databases. I can't recall the exact levels of doses that were used in those databases, but I think if we refer to those, we can see the results. And because like the results are only as good as the data sets that were used to train the models, what I do recall is that for the NCTR liver cancer database, which was used to develop the DeepCarc, they had their decisions were either 1 or 0, so like either cancerous or not cancerous. And so I think that's one of the reasons why we chose that database, because it was relatively easier to work with, having a single call for each compound instead of like a quantitative value for each compound. So I think that may be a limitation of the work that we have done, but in further research, we can try applying these models to different databases that may provide more information on levels of toxicity rather than a single call of like carcinogenic or not.
Yeah, that's a great. You know, often when you're developing a model or a new approach, you start with identifying hazard, and then as you get more sophisticated, right, you can try to build in more elements of complexity. So that's a great way to do it. We have a couple more questions here for you. One of them is, how were the models validated? And what was the predictive power for known carcinogens?
That's a great question. So the models, this is kind of another follow-up from what I just answered, but the models were developed on the NCTR liver cancer database and the DILIST. These databases are relatively small. The NCTR liver cancer database is around 853 chemicals after a bit of pre-processing and getting rid of like something like the organometallics and different compounds that make the deep learning models training difficult. And the DILIS database was around 1,002 chemicals after pre-processing. And so that may be another weakness as well, that the data sets are small. But based on the predictive power, I don't remember the exact numbers, but the F1 score was, I think, 0.9 and higher, or higher for both for both models, and these outperformed QSAR models and state-of-the-art ensemble methods that were trained on the same data sets as well. If you, I can send you the paper for the DeepCarc and Deep DILI models, and they list the exact numerical performance models for Matthews MCC and F1 and accuracy and all those different numbers as well.
That would be great. And are they publicly, are they open?
Yes, they're publicly available.
We put them up on the webinar page, right, with the recording and the papers there. That's great.
And as listed in the presentation, the GitHub repos for these models are all open access as well, so you can access them freely and use them as you wish.
Great. One last question. Can DeepCarc differentiate between genotox and non-genotoxic carcinogens?
I think again, this goes back to the data that it was trained on. I know I keep saying this, but the model is only as good as the data that it's given. And so how well the original data set differentiates between genotoxicity and non-genotoxicity will determine how well the model will be able to differentiate between the two. The good news is the model can be applied to any data set, and I think that's the beauty of deep learning. The model will continue to learn and progress with the data that it's given. The ones we have chosen right now were chosen mainly out of simplicity because they have binary calls for each compound. But in the future, we would like to go into more complex data sets like the DSS Tox data set and other data sets as well. But that I'm sorry I couldn't properly answer your question, but as of now, I don't think it has much power to differentiate between the two. But if we apply it to more complex data sets, I think this power could be developed more.
I think that's, and it's not unique to your work, right? There's a lot of models where, in fact, because of the availability of data, we don't have that mechanistic underpinning to be able to say this is, you know, genotoxic or non-genotoxic, and then build that into the model, right? It's I think it's a challenge for a lot of different endeavors.
Yeah.
Another question's come in. There's a lot of interest in your work. Besides log P and molecular weight, what other chemical parameters were used?
We didn't explore any other specific chemical parameters, but we could explore other parts of the Lipinski's rule of 5, like such as how many hydrogen bond donors and acceptors that the molecule may have. I think, and all of these different exploration options are provided with the ICE, the Integrated Chemical Environments tool, developed by the DNTP. And so that's another area of research we can look into. But if I recall correctly, I think the most interesting part of like the chemical characteristics of the compounds were from the molecular weight and the octanol to water partition coefficient. And so that's why we tried to highlight that in the presentation. But I'm sure with more analysis, there could be other interesting chemical differences that could be found. But yeah, going beyond the Lipinski's rule of 5, yeah, so.
Okay. And one last question, and I wonder if it's maybe more of a suggestion or consideration for future work, but thinking it's in the chat in case you want to look at it, but it's wondering if you've had any thoughts about species differences. So either between rats and mice, right? Because cancer studies are conducted in both rats and mice usually, or differences between rats and mice and humans, and how to kind of integrate that into the modeling.
I think in the beginning of the presentation, we mentioned conventional toxicity testing relies heavily on animal models, and those generally result in flawed extrapolations to humans. And most of the data that we work with are from these animal models. And so it could be the case that some of the results that we get from our deep learning also may have flawed extrapolations as well. Sorry, I kind of forgot what the question was.
About species differences. Like if data is coming from mice or rats, and then, right, as you say, there is that challenge, right, of extrapolating from either mice or rats to humans.
Yeah, I don't know, species differences, I think maybe more of a biologist would be able to understand it better, but for now, I'm working with like the data sets that are given to me. So I don't think I have much expertise about the species differences. So I'm sorry I couldn't answer your question.
No, yeah, I think, you're right. We work in these teams, right? And toxicology is becoming a lot more multidisciplinary. We need engineers and biologists and toxicologists and molecular and cell biologists. And so I think that's a benefit of your project for sure. Okay, well, I think I will close the webinar. We're out of questions, and thank you all for your presentation, for your attention as an audience, but thank you both to Amber and Sunggun for your talks, for sharing your work with us, and we'll see you next month. Bye-bye.